In this third part of the video lecture series, so we will be discussing about the mitochondria, the chloroplast, and the cytoskeletal elements. So the mitochondria, or singularly the mitochondrion, is the eukaryotic organelle that makes the energy molecule ATP through aerobic respirations. So it is a double membrane system. So that means it has two membranes. You have an inner and an outer membrane. The inner membrane is larger in terms of surface area than the outer membrane. So the buildup of hydrogen ions in the outer compartment drives ATP synthesis. So that's actually the electron transport chain is the one that drives the formation of ATP. So it has its own DNA and ribosomes. So the mitochondrion itself is its own genetic material. So in fact, the fact that it has its own genetic material as well as a ribosome, the mitochondrion is thought to be an ancient bacteria that was swallowed up by a proto-eukaryotic cell way before, and then it evolves into the organelle we know now. So the mitochondrion greatly resembles bacteria, and that's one of the theory of its formation. So you have this is an electron micrograph of your mitochondrion. So this is from a pancreas of a bat. So as you can see, the lines, these lines here, are basically the folds of the inner membrane. So you have a larger inner membrane as opposed to a smaller outer membrane. So you have a double layer of a membrane. So that the inner compartment and then the outer compartment. So that is your mitochondria. Now we have the next one is chloroplast. Chloroplast is not found on all eukaryotic cells. In fact, they are only found on specialized cells specialized for photosynthesis. So plastids are organelles that function in photosynthesis or storage in plants and algae. So under them we have the chloroplast, which is the specialized organelle for photosynthesis. So they are the ones that create and they store pigments like chlorophyll. Actually, we have two types, chlorophyll A and chlorophyll B, and also we have other colors, other pigments. Amyloplasts are organelles that make and store starch grains. So this is a chloroplast, packed cells. So this is a cell of a leaf in a flowering plant, and the green ones, those are chloroplasts. So there are a lot of chloroplasts in the plant cell. So this is your interior structure of the chloroplast. This is a TEM image. So you can see here the dark bands. They are basically your thylakoid inner membrane system. So like mitochondria, they also have inner membranes. So they resemble actually pancakes. So and then you have the stroma, which is the interior of the jelly-like interior of your chloroplast. So the next part is the cytoskeleton. So the cytoskeleton is an interconnected system of many protein filaments. So some of them are permanent, some of them are temporary. So the cytoskeleton helps reinforce, organize, and move cell structures or even the whole cells. So it's like the bones, your own bones and muscles. So here is a fluorescence micrograph of the cytoskeleton of a nerve cell. So the yellow color, the yellow dye binds to the cytoskeletal elements, and you have here, this is the concentration of your cytoskeletal elements. So we have three cytoskeletal elements. We have the microtubules, the microfilaments, and the intermediate filaments. So the microtubules, they are like their name suggests, it's tubular. So they are long, hollow cylinders made up of tubulin. So they are dynamic scaffold. They form dynamic scaffolding. So when we say dynamic scaffolding, they can be disintegrated or formed at will. So at one time, we have microtubules forming on one part of the cell, and another part of the cell, you have microtubules that are disintegrating. And then we also have the microfilaments. They are mostly made up of globular protein actin and make up your cell cortex. And then we also have intermediate filaments, which is used for maintaining the cell and tissue structures. So these are the three elements, the cytoskeletal elements. So this is the tubulin. So the tubulin, the one subunit of tubulin is not exactly a tube. Rather, the tubulin helps form a hollow tubular structure to form your microtubule. This is your microfilament, made up of the actin subunit. So the actin, they form somewhat like helical ropes or bands. So that's your microfilaments. And then this is your intermediate filament. So slightly thicker than your microfilaments. And you actually, this is in this form, your microfilaments can also be elastic. So they form called sheets. So these are sheets of tetramers of your dimeric molecule. So that's your cytoskeletal elements. So in closely working with the cytoskeleton is are the motor proteins. So motor proteins are accessory proteins that move molecules through cells on tracks of microtubules and microfilaments. They are able to move because of the energy provided by ATP or adenosine triphosphate. Example of that are the kinesins. So this is a kinesin. So which is the kinesin? This particular molecule is the kinesin. So the kinesin is moving along the microtubule. So it's basically walking, and then this is your vesicle that it's carrying. So it's used to transport vesicle, and they walk along tracks of microtubules. So that is those are your motor proteins. So another subpart of the cytoskeleton are the cilia and the flagella. So they are formed from microtubules that are organized into arrays, 9+2 arrays. So cilia are usually the short hair-like structures in the surface of your cells, and while the flagella, they are the longer whip-like structures. So microtubules grow from a barrel-shaped centrioles, which remain in cytoplasm below the basal body. So basically, this is a cross-sectional view of your microtubule. So if you cut your flagellum or your cilia in half, so you will see this one. It's enclosed in a plasma membrane, and then these are the microtubules forming a circular ring around a certain motor. So this is the flagellum. So this is a stem cell, or rather a sperm cell. So you have your flagellum. So it's used for movement or locomotion. So this is your cilia. So the ciliary structure, they are actually made up of arrays of your microtubules. So there are many ciliary structures on the surface of your cell. So pseudopods or false feet, they are temporary irregular lobes formed by amoebas and some eukaryotic cells. So they are protrusions in the cell. So when the cell wants to move, they can form protrusions. The protrusions are formed because cytoskeletal elements such as the microtubules extend to the outside, and it stretches the membrane, so you form false feet. So they bulge outward to move the cell or engulf your prey. So elongating microfilaments forms the lobe to advance in a steady direction. So you have motor proteins attached in the microfilaments to drag the plasma membrane along. So these are examples of pseudopods. So basically, if this is your cell, and then your cell will form a microtubule, so it will stretch this membrane, you will form feet-like protrusions on the actual cell body. So that is your pseudopods. Okay, so that's it for now. So the next part, the next and the last part of this lecture, we will be discussing about the specialized compartments or specialized structures on the cell surface. So stay tuned.